Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing National Entrance Screening Test NEST 2022 Session 1 question paper. I have done only the biology section. So coming to the first question, the circulatory system of an organism is depicted in the schematic below. The organism is. So the diagram here what they have given, it is of double circulation in which the blood passes through the heart twice before completing a full circuit of the body. So here you can see in the figure the blood passing through the heart twice. So blood is pumped from the heart to the lungs, and from the lungs it returns to the heart before being distributed to other organs or tissues of the body. And double circulation, it occurs in mammals, reptiles and birds. So rabbit is the answer. It is a mammal. A student working in a lab had 3 tubes with the following preparations: freshly extracted nucleic acids dissolved in a buffer, chlorophyll extract from spinach leaf, red colored anthocyanin from onion cell. The student used spectrophotometer to determine the wavelength of light at which each preparation would show maximum absorption or absorption maxima. She noted down these values in her notebook. However, when she came to the lab the next day, she realized that she had not mentioned the name of the sample preparation against each value. The values she recorded were 480 nanometer, 260 nanometer and 642 nanometer. The respective values for the samples 1, 2 and 3 would most likely be. So here the student, she had prepared samples of nucleic acid, chlorophyll extract and anthocyanin extract, and then she had taken spectrophotometric reading. But she did not write the name of the sample preparation against each value. But she recorded the samples at 480 nanometers, 260 nanometers and 642 nanometers. So what are the respective values for the samples 1, 2 and 3 would most likely be? So nucleic acid has absorption value at 260 nanometers. So first will be 260 nanometers. Chlorophyll has got absorption value at 642 nanometers. So second will be 642 nanometers. And anthocyanin is at 480 nanometers. So third will be 480 nanometers. So the answer is option 2. Then next one, an experimenter intends to develop a small molecule drug for inducing polyploidy in a crop plant species. The stage of the cell cycle that such a molecule should affect is. So polyploidy is a condition in which cells of an organism have more than one pair of chromosomes. And the formation of polyploidy is due to the failure of the chromosome pairs to separate during the anaphase of mitosis. This will result in 2 sets of chromosome in a single cell, or I can say that it leads to the formation of a polyploid cell. So here they have asked the question, the stage of the cell cycle that such a molecule should affect is. That is, the molecule inducing polyploidy will affect at the anaphase stage. So the answer here is option 3. An organism M has physiological features as listed in the table below. Mode of nutrition, photoautotrophic, respiration, anaerobic, photosynthetic parameters, sulfur as electron donor and generates NADPH, photophosphorylation, non-cyclic, and carbon source is carbon dioxide. So from the options we can see that algae and cyanobacteria, they both are aerobic. So we will come to green sulfur bacteria or the green bacteria. It is anaerobic, it is photoautotrophic, and sulfur is acting as electron donor and it is non-cyclic photophosphorylation, and carbon source is carbon dioxide. So the answer is option 1, green sulfur bacteria or green bacteria. Then, if the transcription unit of a gene has its promoter and terminator arranged as shown below, then the sequence of mRNA transcribed from this DNA would be. So, promoter sequence, they define the direction of transcription and indicate which strand will be transcribed. Moreover, promoter region contains recognition site for RNA polymerase. So, it is at this region that the RNA polymerase will bind, and it will start synthesizing the mRNA in this direction, that is 5' to 3' direction. So the first Since it is mRNA, instead of T, you will have to use U. So here it will be A, U, G, C, U, A, U, U, G, U, A, U, C, G, C, A, U. That is in the 5' to 3' direction. That is this direction. I have transcribed the strand of the DNA to mRNA. Now they have asked to write in the 5', 3' direction. So it has to be written from here. From this direction you have to write 5' to 3'. So it is AUG, CU, AU, UU, UGU, AUC, GC, AU. So that is the correct option. And here these two are not correct. You have written it from the 3', 5' direction. So they are not correct. Then here, even though it starts with AUG, this codon is not correct. CGA. It has to be CUA. It is not CGA. So that is not correct. So the answer is option 2. Then next one. A histogram of the net primary productivity of selected ecosystems M and N and O is presented below. M and N most likely represent. So M, the highest net primary productivity occurs in tropical rainforest. So N has to be tropical rainforest. And the lowest occur in the deserts. So M has to be the desert. Now third one we will see from the options. So in the option you will get M as desert, tropical rainforest as N, and O as cold deciduous forest respectively. So answer is option 4. Okay, next one. A tree depicting the evolutionary relationships between a few animals is given below. Lancelet, lamprey, bass fish, frog, turtle, leopard. The correct statement regarding the characters 1 to 5 is. So characters 3 and 4 are amnion and 4 limbs respectively. 3 and 4. 3. Your frog is not having an amnion, so that is not correct answer. Then next one. Character 4 could be hair covering the skin. Reptiles don't have to turtle over there. So character 4 will not be hair covering the skin. It is not a characteristic of the reptiles. So 4 is also not correct. Then character 1 could represent loss of limbs while 2 would represent lungs. 2 can represent lungs, but lancelet and lamprey, they don't have limbs, so it is not loss of limbs. They don't have limbs itself. Third option is also not correct. The remaining is the fourth option, that is character 2 and 5 could represent hinged jaw and hairs. Yes, 2 can represent hinged jaw, and 5 leopard. It is having hair. It is a mammal. So 2 and 5 characters are correct. So answer is option 4. Then a biochemical reaction is catalyzed by an enzyme E. An experimenter measured the amount of product P formed in the reaction either in an uncatalyzed or a catalyzed reaction, keeping other reaction condition identical. In the uncatalyzed reaction, 19,800 millimoles of P is formed from 20,000 millimoles of R at the end of 1 hour. Continuing the reaction for another 1 hour did not increase the amount of P any further. In the catalyzed reaction, the experimenter added 1 millimole of the enzyme to the reaction. The amount of P expected at the end of 1 hour in the catalyzed reaction is. So here the experimenter, he has conducted 2 experiments. That is, in one he has added the enzyme and in one he has not added the enzyme. He found that in the uncatalyzed reaction, 19,800 millimoles of P is formed from R at the end of 1 hour. And if he continued the reaction for another 1 hour, he did not saw an increase in the amount of P any further. Then in the catalyzed reaction, he added a certain amount of enzyme into the reaction and he is The amount of P expected at the end of 1 hour in the catalyzed reaction is. So actually when you add enzyme to a reaction, it decreases the activation energy for the reaction and increase the rate of reaction. It does not increase the amount of product. So the amount of product remain the same in an enzyme catalyzed reaction also. So the amount of product that you will get in both the catalyzed and uncatalyzed reaction will be 19,800 millimoles. So the answer is option 3. Then, in an experiment, a scientist captured and cut the tails of wild male swallow birds that had already set up territories. He then glued the tail feathers back on these birds to get 3 groups of birds with differing tail length. Tail longer than before, tails shorter than before, tails of the same length as before. These males were then allowed to mate based on the assumption that attractive male birds would require lesser time in days to acquire a mate. The number of days it took to find a mate in each group was recorded. Mean values for each group are shown in the graph below. Based on the observation, the correct statement is. So first option, I have given the graph again also here for explanation. So first option, female birds have higher preference for male birds with short tails. It is not short tails, they have preference for long tails because long tail birds took lesser time to acquire a mate when compared to short tail and control. So they have high preference for long tail birds. Then coming to third option, there is no difference in the time taken for mating in birds with short tails as compared to birds with unchanged tail length. Here unchanged tail length is your control sample. So there is a difference. Control took lesser time than short tail birds. So there is a difference in the time taken for mating in birds with short tail as compared to birds with unchanged tail length. Then fourth option, the presence of tail is more effective in attracting females than the length of the tail. So the length of the tail is more effective in attracting female. The long tails birds took lesser time to acquire the mate. So what is left out? Option 2. That is, a male normally takes around 8 days after arriving in the breeding area to find a mate, as you can see in the case of control. It took around 8 days. So it is less than 10, so it will be 8. So it took around 8 days after arriving in a breeding area to find a mate. So the answer is option 2. Then phytohormones play a crucial role in regulating growth and development of plants. The table below lists a few phytohormones and their physiological response that they regulate. So abscisic acid is leaf fall dormancy, that is first one is S. Then gibberellic acid is internode elongation R. Auxins is apical dominance P. Cytokine is cellular expansion Q. So the answer is option 1. It is just a simple match the following. The next one. Identify the correct statement about thylakoid membrane that make those the sites of light dependent reaction in photosynthesis. Carbon dioxide and water can diffuse through the thylakoid membrane. More correct will be H positive ions can diffuse through the thylakoid membrane. Photosystem 2 anchored to the thylakoid membrane is involved in both cyclic and non-cyclic photophosphorylation. Photosystem 2 is involved in non-cyclic photophosphorylation only, not in cyclic. Then cytochrome complex embedded in the thylakoid membrane creates a proton gradient. That is correct. 3 is correct. Then fourth one, thylakoid membranes with pigments and protein complexes has the capability of harvesting light. That is also correct. So 3 and 4 are correct. Coming to the next question. In a certain population of beetles that is in Hardy-Weinberg equilibrium, black color is recessive phenotype and grey color is dominant phenotype found in heterozygous and homozygous individual. So black color is recessive phenotype and gray color it is found in heterozygous as well as homozygous individuals and it is dominant phenotype. If the percent of individuals with black color and gray color in the population is 9% and 91% respectively, then the percentage of the homozygous dominant individual would be. So they have given Q square, that is 9%, and Q is equal to 0.3. We know from Hardy-Weinberg formula P plus Q is equal to 1 and P is equal to 1 minus Q, that is 1 minus 0.3, that is 0.7. So P square is 0.7 into 0.7, that is 0.49. So the answer is 49%, option 3. Then next one. The purple frog Nasica batrachus sahyadrensis is a species endemic to the western Ghats of India. Another frog species, marble balloon frog Euperodon globulosum, looks very similar and occupies similar habitat as the purple frog. Marble ballooned frog is also endemic to India. Based on these observations, Nasica batrachus sahyadrensis and U. globulosum are thought to be close relatives. A biologist studying the evolution of frogs of the world find out that the closest relative of the purple frog is another frog genus Zooglossus, which is endemic to the islands of Seychelles near Africa rather than the marbled balloon frog. So there are two frog species in India, that is purple frog and marble ballooned frog, and these two frogs they occupy similar habitats and looks also very similar. Now based on these observation, it was told that the purple colored frog and marble frog are close relatives. But a biologist who was studying the evolution of frogs of the world found that the closest relative of the purple frog is another frog belonging to the genus Zooglossus, and it is found in Seychelles near Africa. So, and marble balloon frog, it is not a close relative of the purple frog. Now next para. A naturalist observed that some species of Zooglossus have specialized structures to live on high tree canopies, strongly webbed toes for gliding. Some had strong limbs for burrowing lifestyle, while some lived in fast-flowing streams, and one species of Zooglossus seychellensis lived among the leaf litter of the forest. So, it was observed that the Zooglossus had specialized structures to live on high canopy trees, fast-flowing streams, then burrowing lifestyle, then some had were found to live among the leaf litter of the forest also. So there were different subspecies or species of Zooglossus, different species of Zooglossus with specialized structures. Then next para. The tree communities on the southwestern coast of Seychelles is different from that of the eastern coast. As a result, the color of the leaf litter also varies. The Seychellensis on the eastern coast are black colored, while the same species is light brown colored on the western coast. The population size for the intermediate colored S. seychellensis, that is dark brown, is very small and rarely found. So only two extremes of the species of seychellensis, that is black colored and light brown colored, were observed, whereas the intermediate colored seychellensis, that is dark brown colored, was found to be very small in number or even rarely found. Based on this information, choose the correct interpretations. The genus Zooglossus has undergone adaptive radiation. That is correct, because adaptive radiation if the rapid proliferation of a species result in an array of species that live in a variety of environments and differ in characteristics they use to exploit those environments, the radiation is said to be adaptive. You can see that in the second paragraph of the given paragraph. So in the second para, you can see that the genus Zooglossus has got various adaptive features. That is, some can live in burrows, some can live in leaf litter, some live in fast flowing streams, etc. So that is a type of adaptive radiation found in the genus Zooglossus. The marble balloon frog and the purple frog have undergone convergent evolution. Now, what is convergent evolution? It is the independent development of similar traits or features in unrelated or distantly related species or lineages that typically occupy similar environmental or ecological niches. So the marble balloon frog and the purple frog, they live in the same ecological niche. They have similar features also. So they were once considered to be close relative, but actually they are not closely related. They have undergone convergent evolution. So second option is also correct. The species of Seychellensis is undergoing disruptive selection. So disruptive selection, it produces a population that has two extreme versions of a trait. Such as if you have got short and tall organism, medium height organism will not be favored. That is called as disruptive selection. Here in the third paragraph, you can see that the Seychellensis, it has got two colors. One is black and the one is light brown. But the dark brown, that is intermediate color, that is dark brown, is not favored. Only the black and the brown colored, light brown colored are favored. So that is an example of disruptive selection. The species of S. Seychellensis is undergoing stabilizing selection. That is also not correct. Then next one. In corn, the genotypes C and R results in colored kernels, whereas homozygous recessive condition for at least one of these genes results in colorless kernels. Assuming that C and R are unlinked genes, choose the correct statement. So I have given for each option, that is A, B, C and D, I have mentioned it like that. For each option I have given description of how half colored and half colorless were obtained, or how in some cases I have drawn the Punnett square also. You can go through that and then you will understand. So capital C and capital R result in colored kernels. Small c, capital R, capital C, small r will result in colorless kernels. And these are the options. C and D also, these options I have given. Then here you can see that there are 3/4 colored and 1/4 colorless in the case of B option. Then 1/4 colored and 3/4 colorless in the case of C option. And in the D option it is 3/4 colored and 1/4 colorless. So you can go through that. The next one. In humans, breathing involves cycles of inhalation and exhalation. The events that occur during inhalation is or are. That is expansion of thoracic cavity, decrease in intrapulmonary pressure. Answer is option 3 and 4. It's a direct question. The next one. An experimenter ligated cDNA of p53 gene into the multiple cloning site of vector whose map is shown below. The ligated vector was transformed into E. coli cells and the transformants were initially selected on an agar plate containing antibiotic P. The bacterial colonies obtained on this plate were further propagated sequentially on antibiotic Q and then antibiotic R by replica plating. The correct sequence of P, Q and R to be used to select E. coli colonies with p53 cDNA cloned into the MCS is or R. So here this is the vector that is being used. Then there is the p53 cDNA which has to be cloned into the multiple cloning site of this vector. And from the diagram you can see the multiple cloning site is in the ampicillin resistance gene. So once you clone the DNA, that is p53 gene, into the multiple cloning site, ampicillin resistance gene is getting disrupted. So when you grow this particular recombinant in a medium containing ampicillin, it will not grow. Now they have asked for selection of this particular recombinant. How will you select? So we will see. So chloramphenicol, tetracycline and ampicillin. Tetracycline, chloramphenicol and ampicillin. That is the correct answer. So before coming to that, what is replica plating? So a sterile velvet is pressed on the grown colonies. These cells are transferred from velvet to new plates. Growth on the plate is compared with the master plate. Now, recombinants, I told you it will not grow in ampicillin medium, but it will grow on chloramphenicol and tetracycline media. Now P can be either tetracycline or chloramphenicol. We can either choose to plate them first on chloramphenicol and then on tetracycline, or first on tetracycline and then on chloramphenicol. So first, if you are plating the P on tetracycline medium, then Q should be chloramphenicol and R should be ampicillin. Now if P is chloramphenicol, then Q has to be tetracycline and R has to be ampicillin. Now what is happening here? I have shown only one example. The other is also the same method. Okay. Not example as one particular choice. Tetracycline, chloramphenicol, ampicillin. For chloramphenicol, tetracycline, ampicillin also it is the same method. So first you are plating them on tetracycline medium. This is how you plate it. Then you are plating on chloramphenicol medium. The same bacterial this thing is replica plated onto chloramphenicol medium. Then from there you are replica plating it onto ampicillin medium. Now what you will do is, when you see the ampicillin plate, you can see two colonies missing. So that means those colonies are unable to grow on ampicillin medium, and they are your recombinants. Now if you trace back, you can see that here you have got two colonies, and here also the same two colonies have grown on tetracycline and chloramphenicol medium, which are recombinants. The others, that is these ones, they are non-recombinants. Okay. So the answer is second and fourth. Now next one. In the given pedigree, circles represent females and squares represent males. Filled shapes indicate affected individuals, while unfilled shapes indicate unaffected individuals. Vitamin D resistant rickets exhibits X-linked dominant inheritance. Identify the pedigree from the options that correctly represents the inheritance pattern of vitamin D resistant rickets. Now here the male or the father is affected and the daughters are also affected. So all daughters of the male who has the trait will also have the trait. That is one characteristic of X-linked dominant inheritance. Sons can have the trait only if their mother also has the trait. That is why in third, that is 1, 3, you got a son who is affected by the disease. Since it is X-linked dominant, this daughter also will be affected by the disease, because even if the daughter is a carrier of the disease, she will be affected by the disease because it is a dominant inheritance. X-linked dominant. So answer here is option 1. The other pedigrees are not correct. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.